Okay, so a very good evening to one and all present here. I am Srijan Sen from IIT Kharagpur, and today I will be taking a week 9 live class session on the fundamentals of protein chemistry, which is being taught by Professor Shagota Dasgupta of IIT Kharagpur. So I welcome you all to the week 9 live class session, and today we will talk about the lecture that was taught to you in week 9, and then we will be discussing a few question and answers in the form of assignments. Okay, so let us start with the class then. Okay, so I'm sharing my slides with you all. Okay, so yeah, so today's class will mostly focus on membrane proteins. Right, so previously we talked about a lot of proteins, and also mainly we will talk today about membrane proteins. That's the central topic in understanding how the cells interact with the environment and regulate the molecular traffic. And by the end of this, you would be able to know about the membrane structure, the types of proteins, transport mechanisms, and why we feel it is difficult to purify these membrane proteins. Okay, so mostly we will go through the different models, then the types and functions of membrane proteins, then different transport mechanisms, the key challenges we face in the membrane protein purification, and then in the end we will be doing a few applications and assignments. So okay, when we talk about membrane proteins, the first thing that comes to our mind is the phospholipid bilayer, right? So the phospholipid bilayer is, you have the different types of lipids. You have the lipid. Why do we call it the phospholipid bilayer? You all know about that. What it has glycolipids, it has different oligosaccharides, right? Then there are several integral proteins, like it is embedded inside this PLP bilayer. Then some hydrophobic alpha helices. Then we have the lipid-linked proteins. So these are the phospholipids. You have cholesterol and the peripheral proteins. Now the different models that describes this membrane design is, the first one is the fluid mosaic model and the membrane structure. So this model says that membranes are fluid that is pliable and can easily move. They are very flexible. So that is why it's fluidic in nature, and mosaic because it is made up of different types of what do you call macromolecules, like the lipids, proteins, carbohydrates, and all. So you can see from the diagram over there. So the cell membrane is a selectively permeable barrier. Only certain substances can pass through it, and that is why we predicted, because of the two reasons. So it's selectively permeable, that it can directly go through the membrane. That's the size. It's based on the size and the polarity. So when you look into the gases, right? So the gases, then there are hydrophobic molecules that can pass through it. Ethanol can pass through it. There are several gases that can pass through it. But you have the amino acids. There are some charged molecules, then large polar molecules that cannot pass through it. For example, like the glucose and all. So the cell membrane is only let certain substances in and out of the cell. So it is very important to maintain the cell homeostasis or the balance within the cell. So the phospholipid bilayer is made up of polar heads that face outwards, fatty acid long tails, that is the hydrophobic face, which is mostly inwards. It also contains like cholesterol, which changes the fluidity, stiffens at high temperature, and prevents freezing at low temperature. So that's the water interacting part is the hydrophilic polar head, and the hydrophobic mostly embeds the integral proteins and all, and then you have another layer of hydrophilic heads. So you see over here, so this is cholesterol that is also present within this PLP bilayer, and these are mostly attached to the hydrophilic tails inside towards the hydrophobic region. So there are types of steroids, like you have the lipids that changes the fluidity of the cell membrane. So at higher temperature, you see that it's the membrane stiffens, whereas at lower temperatures, it prevents membrane from freezing. So it has a very vital role to play in this particular thing. So now it is very important to like ask us like why, what would happen to the membrane if cholesterol was absent at low temperature. So if you see from over here, so or at high temperature, what would have happened? So it prevents the membrane from freezing. It's very important. So at lower temperature, if cholesterol would not have been there, it would, the membrane would freeze, and that to lead of the shearing of the membrane, and ultimately it would hamper the cells. Now, it is very important to know about the types and functions of membrane proteins. So, like a very brief overview of the membrane proteins, you have the recognition proteins, that is the proteins that have carbohydrate chains attached. Carbohydrate chains help in cell identification. These are the recognition tags, like the name tags which we have, like we have a name, so these have name tags. Like example is the RBC A, B, then it identification for viruses as foreign materials. Okay, the next one is your receptor proteins. From the diagram you can see over there that proteins receptor means it receives something. So what does it receive? It receives the chemical signals from other cells, such as the nervous system cells or the neurons, that play a very important role in the receptor proteins. The third type is the channel proteins. So the channel proteins, we have heard of aquaporins for water passage of the specific substances. So it is also very important because it is also cell permeable, selectively permeable. So proteins that involved in letting certain substances in and out of the cell. So aquaporins, the common example is that lets water in and out of the cells. The other part is that spans the bilayer, which is the integral proteins. It crosses both the layers of the phospholipid bilayer. So these proteins, if you can understand, is made up of a group of hydrophobic residues and hydrophilic residues, which is generally very common in proteins and all. But these integral proteins are very critical for maintaining their activity across the hydrophilic and the hydrophobic. So the hydrophilic layer, the outside environment versus the inside cellular environment, and most of these are important in passing of, acts as uh, you call them as channels or gates, okay, for the passage of ions, ion channels, and different other substances to maintain the balance or to disrupt something. Okay, then you have the peripheral proteins that are located on just one face of the bilayer, either on the top or the bottom of the phospholipid bilayer. Now, the primary functions of membrane proteins is attachment to the cytoskeletons and extracellular matrix, cellular signaling, different enzymatic activities, transport, intercellular joining, cell-cell recognition, so on and so forth. Now, so like I was telling that integral proteins are very, very important. So there are different types of integral proteins, and we need to know them. We also need to know how does these proteins look like. So let's go one by one. So the first one is type 1. It has a single span, transmembrane span, and the N terminus is the ectodomain, the C terminus is the cytosol. So the cytosol is the material that is so the cytosol is the material that is present inside the cell, right? So the N terminus is the ectoderm you see over here. So you have the ectoderm at the N terminus. So the N terminus is in the ectoderm, and the C terminus is towards the cytosol. Now the second type is just the vice versa of it. That is also single span, where the thing is just reversed, where the N terminus is towards the cytosol and the C terminus is towards the ectoderm. Now, type 3 has multiple spans, so you see the entire thing is type 3, so it moves, first it starts over at certain point and then it coils around. The other flexible region goes through to the ectodomain, then you have a certain portion towards the cytosol and all, and so you have a very zigzag structure. The type 4 is also very similar to type 3, but there are certain breaks and also several different polypeptides assembled to form channels. So these are the different channels, and they play a very important role is to act as ion channels or different voltage gates and all. The type 5 is the lipid linked protein. So you see the yellow region over here, it's lipid linked, and both the for the type 6, both the proteins with both trans domain components and have a lipid anchor. Okay. So now we need to understand the properties of integral proteins, a very, very important protein component in the PLP bilayer. So because of their hydrophobic transmembrane domains, integral proteins are difficult to isolate in a soluble form, and removal of these proteins from the membrane normally requires use of detergents or ionic solvents like ASDS, which denatures the protein, or non-ionic detergent like Triton X100, which generally does not alter the protein's tertiary structure. So the use of mild non-ionic detergents for solubilizing, purifying, and reconstituting functional membrane protein system. So this is the structure of the sodium dodecyl sulfate, and you have the Triton X100 that is present over here. Now, membrane proteins are very difficult to isolate for multiple reasons. Some of them being, you already might be knowing about these, that it's hard to overexpress large quantity functional proteins, difficult to separate the proteins from the membrane lipids. To get the protein into solution for purification, it needs to be solubilized with detergents which might destroy the protein. So we need the protein in its functional, folded, monodispersed form, and many detergents needs to be screened. And also it is hard to crystallize with detergent around the molecule. So in total, we can tell that purification of membrane proteins is quite tricky. We have to use different components and all to purify this. But so some strategies or some simplicities that we can do to purify the membrane protein is that membrane proteins already cover more than 50% of the current drug targets and about 30% of the typical cells proteins. So these are very, very important proteins and have very important functions as well. So solubilization of membrane proteins can be accomplished by using amphiphilic detergents, but preservation of the biological and functional activities can be quite challenging processes, as many membrane proteins are susceptible to denaturation during isolation process. Now, detergents is the only way out, or non-ionic detergents or ionic detergents are very important for that, because detergents are amphipathic in nature and contains a polar group at one end and long hydrophobic carbon chains at the other end. The polar group forms hydrogen bonds with water molecules. The hydrocarbon chain aggregate via hydrophobic interactions. At low concentrations, detergent molecules exist as monomers. When the detergent monomer concentration is increased above a critical concentration, which we call as the critical micelle concentration or the CMC, detergent molecules self-associate to form aggregates known as micelles. So that is why it's known as the critical micelle concentration, which is a very important parameter for selecting an appropriate detergent. So it's not like that we can select any and every detergent. We have to use it at a particular concentration to actually make the protein work. And the effective CMC of a detergent can also be affected by other components of the biological system, such as lipids, proteins, pH, ionic strength, and temperature of the medium. The other important thing is about the aggregation number. So this number is the number of monomeric detergent molecules contained in a single micelle. So for the transport of materials, you have the passive transport and the energy consuming transport. So passive transport can happen through simple diffusion, facilitated diffusion, osmosis. Okay, and then you have the energy consuming transport, that is the active transport, and exo or endocytosis. So the first one is diffusion. So we know the movement of molecules in solution from a high concentration of that molecule in the direction of the low concentration of that molecule until that equilibrium is reached. So that is simple diffusion. A drop of dye is placed in water. You see the drop of dye dispersing or diffusing. We say diffusing. So it spreads. So the water molecules occupy that particular space, and you will see that the dye molecules are evenly dispersed. So that is how, so over here you have a high concentration of dye and there is a low concentration of dye around. So it disperses and occupies the space of the water molecules. The water molecules occupy this bit of space and they equally distribute till an equilibrium is reached. So similarly, diffusion happens across a membrane. So if the number of ions or concentration of the salts or the ions are very much inside the cytosol, it goes out, or if it is very less in the cytosol, it brings like O2, CO2, then some calcium, sodium, magnesium, sodium ions. So there is a sodium potassium balance within this cell, and that is maintained through diffusion. So why does passive diffusion through the lipid layer only occur with gases and small lipid molecules? So basically these substances are polar, and so is the lipid layer. The other thing is facilitated diffusion. So why are channel proteins necessary to move hydrophilic ions through the membrane? It's very simple, or you can tell it like proteins form a hydrophilic channel. So ions are charged and they cannot go through the non-polar lipid layer because these are hydrophobic in nature. So passive transport by facilitated diffusion happens like there are carrier proteins that allow as a binding site for a specific molecule. Then this can be amino acid, sugar, small proteins that can come and go through these channel proteins. So the molecule enters the binding site. The carrier protein changes the shape, transporting the molecules across the membrane. And when you see that there is one gate that opens, there is the other gate that also closes. So you have the extracellular fluid containing of so many important things. If they have to get an entry, the door opens, the other door closes, and there is one entry at a time. So it's like going into the cinema hall. It's like checking out the tickets and like going inside. So the carrier proteins resumes the original shape after the molecule has entered. The other phenomenon is very common, similar to diffusion, and we all have studied about this, that is osmosis. So the diffusion of water through a semi-permeable membrane. So water moves from its own high concentration to its own low concentration through a semi-permeable membrane. So in which direction will the water move over here? So it moves from a higher to a lower concentration. But what's the difference between diffusion is that it has to pass through a semi-permeable membrane. Now, now up to this is what it does not require any sort of an energy or something. Now, active transport is an energy requiring process where there is need of energy in the form of ATP. So movement across a concentration gradient. So you have a proton pump over here. So proton pumps, endocytosis, exocytosis, that's for bulk transport, then protein pumps for molecular transport. So these so these processes occurring through these protein pumps, endocytosis, exocytosis, is via the active transport. So active transport is the movement against the concentration gradient. So you have to put up some kind of energy or provide some kind of energy such that that movement or can happen from say a low to a higher concentration. That requires energy in the form of ATP, allows for stockpiling, and it's a one directional movement. Oops. Right. So, okay. So, like maybe a few things that I may tell, because this is all about the chapter. I do not have anything much else to tell. So, what you need to know is like why are membrane proteins hard to study? Because hydrophobic regions make them difficult to solubilize. Then detergents are needed, but some denature the protein. Even some proteins are so much sensitive that even mild detergents must be carefully chosen. So proteins need to be functional and monodispersed for this study, and also crystallizing these proteins for structural studies is quite challenging. Therefore, purification is a big challenge. And so why are biological implications of improper folding or denaturation during protein like that? You can answer basically when you look into the detergent concentration. So as I was saying, that detergents are amphipathic in nature, so that means they have both hydrophilic and hydrophobic regions. So at higher concentrations they form micelles, that is the critical micelle concentration, and CMC depends on lipids, proteins, pH, ionic strength, temperature. And membrane proteins are like present in most of these current drug targets. We all know about it. And then like cellular proteins are about 30% of the cellular proteins are membrane proteins, and problems with this transport or recognition are linked to diseases like the cystic fibrosis, then Alzheimer's, so on and so forth. Okay. So this is all about the membrane proteins and the study of membrane proteins. With this idea in mind, we should be able to solve this particular problems. Okay. So like, okay, so maybe if you guys have any questions, you can ask. I'll just wait for 5 minutes and then we will start with the practice problems. In the meantime, if you have any questions, feel free to ask me. Also, if you have any questions from the previous classes, you can go ahead. So I guess you do not have any questions. So we are good to go with the assignment then. The practice problems. Okay. The first one says identify the region. So you people can give me an answer and then I will tell you. Okay. So the first one is identify the regions marked X and Y. You can write in the chat box.
Integral protein, then peripheral protein.
Sorry. I did not get you actually. You can write it in the chat box if that's not a problem. You can tell me, but that is also fine. Absolutely. So X is an integral protein. That is correct. Y is the peripheral protein. That is absolutely correct. Option A. Good. The next one says which of the following motif is mentioned in the above figure. So 1A is correct. The next one is which of the following motif is mentioned in the above figure. So what do you see from over here? Is it an alpha helix motif or an alpha helical bundle, beta sheet, beta helix? What does it look like? Alpha helical bundle. So if you see over here, it's very much rich in beta sheets. Okay. But we do see a few alpha helices also over here. So there is an alpha helix over here. You see there is a 310 helix over here. There is also another a little bigger than 310 helix, another alpha helix over here. So we have three alpha helices very much rich in beta sheets. So we can, so calling it an alpha helical bundle won't be that correct. Rather, we can call it a beta helix instead. So question two, that is which of the following motif is mentioned? The answer will be beta helix. That is option D. Okay, right. The next one says, so this is a sequence of amino acids that is given to you, MLF, YBW. So that's a sequence that is present in the dash of globular protein. Interior hydrophilic core, interior hydrophobic core, exterior hydrophobic or exterior hydrophilic surface. Okay, so which one is it? Is it the interior hydrophilic or hydrophobic core or the exterior hydrophilic or hydrophobic core? So which one of the options do you think is more appropriate? So you have to study basically the amino acids. So in case if you are not aware of the short terms of this, it is methionine followed by leucine, phenylalanine, tyrosine, valine, and tryptophan. So,
Tryptophan.
huh?
Yes, sir, tryptophan.
I'm not getting you. Your voice is not that clear. Like option. So if you look into the amino acid sequence and all, you can understand that it is a very hydrophobic rich sequence. Now, to be on the surface, there has to be a few hydrophilic residues at the N or the C terminus. But if you see, the N and the C terminus is so occupied with the non-polar regions and so on. So therefore, are some like non-polar moieties. That is why it should be present in the core. Now, being hydrophobic rich, the interior. So the right option is interior hydrophobic core. It has to be in the core because on the surface, if it has to be on the surface, it's either ends have to be hydrophilic, and then it can go. And anyways, the sequence is rich in hydrophobicity. So that is why option B is correct. The next one says choose the incorrect statement about Gramicidin A. It works as an ion channel type ionophore. It is a 15 residue linear peptide. It's a ribosomal protein. It consists of alternating D hydrophilic and L hydrophilic amino acids. So the so what is like choose the incorrect statement about Gramicidin A. So what is Gramicidin A? So Gramicidin A is a well-known antibiotic that forms a channel in lipid bilayers, allowing the passage of monovalent cations like sodium and potassium. You know that about it is composed of 15 amino acid residues, although it forms a helical structure within the membrane. Then Primethicillin A is definitely composed of D-hydrophobic and L-hydrophilic amino acids, and these amino acids are contributing to its ability to insert into the lipid bilayer. It does not consist of any alternating hydrophobic or hydrophilic residues in a way that would make it amphipathic in the conventional sense of membrane insertion. So number 1, 2, and 4 are the correct options over here. Well, the incorrect option is it is a ribosomal protein. It's not a ribosomal protein. Okay, it's an antibiotic of in more precise manner. So the correct option is number 3 only. So option E. What? Which of the following types of ATPase undergoes reversible phosphorylation to transport cations across the membrane? Which of the following types of ATPase undergoes reversible phosphorylation?
Sir, is it P-type ATPase?
Right. So P-type ATPases are known for the reversible phosphorylation during their catalytic cycle. That is crucial for the active transport of cations like sodium. Then you have potassium, calcium, hydrogen across the biological membranes, and so this phosphorylation involves. So how does this occur? So there is a aspartate residue in the particular enzyme, and the phosphorylation involves this conserved aspartate or aspartic acid residue in this ATPase. Okay, so right, so P type is the correct answer. Very good. The next one says bacterial porins are beta barrels. Which amino acids would you expect to be particularly abundant at the entrance of a beta barrel that is specific for phosphatides? So there is the entry of phosphatides. So you need some amino acids to be present at the gate, like some
Lysine and arginine.
fantastic, because they are positively charged, and the
Yes,
physiological
sir.
pH are also ideal for them to interact with and facilitate entry of the
Hmm.
phosphate ions. So what they do to they attract and bind to these negatively charged ions and allows them an entry. So option A is correct. The next one says all aerobic organisms require dash to produce the basic energy for by generating ATP by an ATP synthesizing enzyme called dash through a process called dash transport. So too many of things. So you have to answer the one, two, and three. What is one, two, and three?
All aerobic organisms require oxidative phosphorylation to produce the basic energy by generating ATP by an ATP synthesizing enzyme called, is it ATP synthesis, sir?
Right. And yeah, you're going correct. So first one is oxidative phosphorylation, then it is ATP synthase. The next one.
So process is called um, electron transport, sir.
Right, right, right. So it's an electron transport. Very good. So that is option A. So oxidative phosphorylation to produce ATP by ATP synthase through electron transport. Correct. Now the next one is Soret band is mostly due to dash transition. It is useful to characterize the dash prosthetic group. So what is a Soret band? So if I like tell you about Soret band, so it is a characteristic absorption band in visible spectrum for many porphyrin containing compounds due to a certain transition which you need to tell me. So there is a certain transition occurring within the porphyrin ring, and it is also so it is prominent in a Prosthetic
Heme
group.
transport, sir. Pi to pi star.
Pi to pi star, very good. So there is a pi to pi star transition that is happening
Heme
within the
group.
porphyrin ring.
Heme prosthetic group.
Right.
Yeah.
So it is a characteristic of this heme prosthetic group, which is also a porphyrin derivative found in hemoglobin cytochromes. So option C is the correct one. Right. The next one says indicate whether the following compounds are likely to cross a membrane by channel or transporter. So whether they will cross through the channel or the transporter, calcium, water, glycine and glucose. And how does a channel work and a transporter work? How are they different? So yeah, we are learning quite a few things from these questions as well, which is great. So channel versus transporters. So maybe a little bit about channels and transporters. So channels, it it functions on the principle of diffusion, if you know that. So it facilitates diffusion of small polar molecules down the concentration gradient, sometimes ions and all. So they have to cross through the channels that are uncharged polar, whereas transporters also, which we know as the carriers. So from the name, if you can understand that it binds to certain molecules, and then they have to undergo certain conformational changes to move across the membrane, and then there is an involvement of a certain type of transport, whether it can be an active transport or a passive transport. So these require the transporters to cross the membrane due to there's either size constraint or specific recognition requirements. There is a change in the shape and the size, and only then they get an entry. Right. So the process of a channel is more simple. For a transporter, there are so many structural changes that are happening, binding and all, and then it goes. So from the things that you can see over here, like calcium, water, glycine and glucose, which of them will pass through the channel and which of them through the transporter? So the polar molecules over here are what? You have the ions, you have water. So 1 and 2, that is calcium and water, will pass through the channel. And then glycine and glucose, these are molecules,
Glycine and glucose process
they
to
have only
transporter.
passed through the transporter. So option E. The next one says at 37 degrees, the internal cell concentration of sodium is 20 millimolar and the external concentration is 340 millimolar. So you have to calculate the membrane potential required to transport sodium out of the cell. How do you do it? What is the equation that you are going to use? Okay, maybe I will just try to solve it. So you have delta G, that is 0. You have delta E, what is given over here? So delta E is RT, then ln. You have concentration outside, yes. So like okay. Concentration of Na out by concentration of Na inside divided by Zf. What is Z? That is the charge on the ion. So that is 1. So you can neglect that, and if you calculate this, it will be somewhat around like so the temperature over here is, sorry, T is like 37, so 310 Kelvin, and R you know, so delta E is equal to minus. Okay, if you calculate this, you will get that particular answer. So option A is correct, right? Great. So the next one says, if the substance is uncharged, what information do you need to predict whether the transport will be thermodynamically favorable? If the substance is uncharged, what information would you need to predict whether the transport will be thermodynamically favorable? So for an uncharged substance, its movement across the membrane is mostly driven by the concentration gradient,
Concentration gradient.
and the substance will therefore move from a higher concentration grade towards a lower concentration, because that is the more thermodynamically favorable process due to delta G being negative. So there is a downhill transport. The pH gradient and the membrane potential are more relevant for charged substances or processes that are coupled to the proton movement. Okay, and you do not need about solubility, that's for non-polar core of the membrane, its ability to cross the membrane, but not the thermodynamic favorability of the overall transport, okay, across it. So yeah, so therefore the concentration gradient would be the driving force when it's an uncharged molecule, right? So option D. The transfer of electrons from NADH to coenzyme Q is hindered by X when it binds to iron sulfur proteins. Blocking the energy transport, Y inhibits the activity of cytochrome oxidase, and Z binds to the alpha subunit of Na plus K plus ATPase and inhibits its activity. So you have to find out what is X, Y, and Z. So the first one is something basically hinders this NADH to coenzyme conversion. Okay, so what will be that? Then is something blocks the electron transport and the activity of cytochrome oxidase, and there is a third thing that binds to the alpha subunit of Na plus K plus ATPase and inhibits its activity. So, so the first thing happens in complex 1, where rotenone is a known component for inhibiting the transfer of electrons from NADH to coenzyme Q. Carbon monoxide, it's a very nice or a very hazardous gas that also blocks the electron transport and inhibits the activity of cytochrome oxidase. And then there are some cardiotonic steroids that binds to alpha subunits and of plus, okay, of Na plus K plus ATPase and inhibits its activity as well. So option B is the correct one. Okay. The next one. So classify the types of solution as isotonic, hypotonic, and hypertonic. So A, B, and C. So which is hypotonic, which is isotonic, and which is hypertonic? What is your answer? So what are the meanings of these three terms? What do you mean by hypotonic, isotonic, and hypertonic? So hypotonic solution, it is a solution with a lower solute concentration than the cell's cytoplasm. So water moves into the cell, causing it to swell or even burst. So that is lysis. So that is a hypotonic situation. So which of them do you feel is that? It is option B, because it is so filled up that it is going to burst. So B is hypotonic. Isotonic, so it is a solution with the same solute concentration as the cell's cytoplasm. So there is no net movement of water in or out of the cell, and the cell maintains its normal shape and condition. So that is option C, isotonic. There is no movement. Water comes in, water goes out. And the third one is hypertonic. So it's a solution with higher solute concentration than the cell cytoplasm. So water moves out of the cell, causing it to shrink. Okay, so that is option A. So that is A. So A is hypertonic, B is hypotonic, and C is isotonic. So option C is the correct one. And the last question over here is, which of the following statements is or are indicative of passive transport? So there are so many options, sentences, statements that are provided. Which of them do you feel is appropriate for passive transport? You want to try answering it? Maybe. Okay. You have said C. Let us find out what is it. So what is basically passive transport? So passive transport, like I said, is a membrane transport that does not require ATP to move. So there is not even like, so instead it relies on the concentration gradient of the molecules. So the first option, that is ATP is required for transport, is incorrect, because ATP is required for an active transport, not for passive transport. The second sentence says small organic molecules will diffuse based on molecular weight and solubility in lipids. That is absolutely correct, because it allows the diffusion of small lipid molecules across the cell membrane down the concentration gradient, and their ability to cross the membrane is mostly influenced by their molecular weight, solubility, and like things like that. Then this third statement is also incorrect, because transport from low to high concentration requires energy, and it is more of a characteristic for active transport. And so the next one is definitely correct, because there is a transport from a higher to a lower concentration across the concentration gradient. So option 2 and 4 are the correct ones. That is option C. Yeah. So right. Okay. So that's all I have from my side. If you have any questions, you can ask me. Any questions like from the previous classes and all. If not, then that's all. I will maybe wait for another 2-3 minutes if there is a question from anyone. So, so this class is anyways being recorded, and if you want to get back and watch over the class, you can see that in YouTube. It will be uploaded over there, and also the NPTEL team would be sharing with you the slides and the recordings. And probably in a few weeks time, or maybe in a month, guys will have an exam. So start preparing for the exam. Go through the assignments and the practice problems that is being taught to you, because these are the things from where the questions will come, very similar questions from this. And if you're giving an exam, it will be very very helpful for you people as well. So go through this and make sure that you start preparing early. Okay, so no questions then. Okay, so let's call it a day. Okay, goodbye. Thank you very much for joining. We
Thank you,
will
sir.
meet in the next week, Friday at 6 p.m.